Welcome everyone to the 2023 PI Conference. My name is Emma Mertens and I will be your moderator for today's session, Gene Therapy and Genetic Testing. Dr. Kuo could not be here today in person, but has graciously filmed today's content. At the end of the presentation, we will also include a Q&A portion based on the most common questions IDF receives regarding gene therapy and genetic testing. If you have additional questions that do not get addressed in today's session, we also encourage you to visit our Ask IDF platform, and we'd be happy to follow up with you individually. Today's session is sponsored by Pharming. We are so grateful for their ongoing support of our education and learning opportunities. Caroline Kuo is an assistant professor of pediatrics in the Division of Allergy, Immunology, and Rheumatology at UCLA. Dr. Kuo attended medical school at University of Southern California Keck School of Medicine and completed her pediatrics residency at Harbor-UCLA. She completed her allergy and immunology fellowship at UCLA and is board certified in allergy and immunology. Her work focuses on the diagnosis and treatment of both pediatric and adult patients with primary immune deficiencies. Her research focuses on novel therapies for rare immunologic diseases, and she is an investigator on gene therapy trials for primary immune deficiencies at UCLA. Welcome, Dr. Kuo.
Hi, everyone. Thank you so much for joining today. My name is Caroline Kuo. I'm a immunologist at UCLA, and today I'll be speaking on gene therapy for inborn errors of immunity. I have no disclosures. These are some of the main objectives of this talk: to understand the curative nature of hematopoietic stem cell gene therapy with autologous transplantation for inborn errors of immunity, know the current gene therapy trials available for immune deficiency, distinguish vector-mediated gene therapy from site-specific gene editing, identify the benefits and risks of gene therapy versus gene editing, and understand some of the basic challenges in the field of gene editing. So as many of you know, a hematopoietic stem cell transplant, or what we often kind of call a bone marrow transplant, is curative for many inborn errors of immunity. And actually, some of the earliest cases in which bone marrow transplant was applied was for primary immune deficiencies. And so what I show here is a paper from the Lancet in the 1960s reporting on a young boy with sex-linked lymphopenic immunological deficiency who underwent bone marrow transplant successfully. This boy ended up having X-linked SCID, but nowadays allogeneic hematopoietic stem cell transplant is being applied for many other inborn errors of immunity, such as SCID, Wiskott-Aldrich syndrome, chronic granulomatous disease, and many others, too many really for me to list here. And the reason allogeneic bone marrow transplant is successful is really well illustrated by this figure of a hematopoietic tree. And what this is showing us is that all of us have multipotent hematopoietic stem cells, which are long-lived, residing in our bone marrow. And these multipotent stem cells are able to give rise to all of the cells in our blood. So that includes immune cells as well as red blood cells. And so if you can replace the hematopoietic stem cells of an individual who has an immune defect with that from a healthy individual, you could essentially replace their immune system. However, the major limitation of an allogeneic stem cell transplant is the unwanted immune responses between a patient and transplanted cells. So a patient can reject transplanted hematopoietic stem cells, or there can be some donor T cells that are contaminating the marrow graft, and it can react against the recipient in something called graft versus host disease. And so many times when it comes to transplant, we are dealing with balancing this phenomena of graft versus host disease and rejection in the recipient. And so thus was born the idea of autologous transplant with gene therapy. And the way that this works is that if you know the gene that is responsible for an individual's disease, and you know the normal sequence of that gene, you could potentially introduce that corrective gene cassette or corrective gene sequence to a patient's own hematopoietic stem cells. These stem cells, because it's their own, could then engraft and pass on that therapeutic gene to all downstream lineages, so also reconstituting the immune system. And so the main hypothesis behind this is that gene therapy using autologous hematopoietic stem cells that are corrected with the normal gene will have beneficial effects on blood cell production or function without the immunologic complications of allogeneic hematopoietic stem cell transplant. And so how this practically would look like is shown in the schematic in the bottom. We're going to focus on the circle that says ex vivo because the field is not quite ready for in vivo gene therapy, even though that's something we're working towards. So what ex vivo gene therapy means is we collect cells from a patient. So we collect bone marrow or peripheral blood stem cells from a patient, and we modify them in a lab before we give them back. So these stem cells that we collect are purified and then put into a short pre-stimulation culture, after which they are then gene modified, so either through gene addition or gene editing. And nowadays, we're able to viably cryopreserve, so we can freeze these gene modified cells without killing them and hold on to them until our gene therapy product is fully certified before we bring a patient back for conditioning and transplant. The benefits of this is that everyone would have a donor because it's themselves. Autologous cells should not be immunogenic. There should be no rejection or graft versus host disease, and immune suppression would not be needed with conditioning or GVHD prophylaxis and treatment. And so I thought I would include in here the importance of genetic testing because it so closely relates to gene therapy and are, enabling more research in gene therapy to occur. So just a couple of general points about genetic testing is that if we can identify a gene that's responsible for a patient's immune deficiency, we can provide genotype-specific management. There's many cases in which we now have specific medications that are indicated for a very specific genetic disorder. Or if we think more broadly, this could mean the difference between closely observing somebody and seeing if their immune system changes over time versus very quickly putting them into more definitive and potentially riskier treatments, such as bone marrow transplant or thymic transplant or anything else that could result in higher rates of mortality. We're also then able to provide genotype-specific counseling. So this includes anticipatory guidance. So we know what things to look out for and also what things to avoid. So for example, some immune deficiencies are associated with radiation sensitivity. And so in those cases, it's important to know if that's the case so that we can avoid excessive exposure to radiation unless it's really needed. And then lastly, I include here that it enables further research. It helps us understand the disease processes more, and also it enables the development of new therapies, and this includes gene therapy. Because as you can tell, if we don't know the gene that is responsible for a disease, we cannot develop gene therapy for it. So typically, the way that we deliver this therapeutic DNA has been through viral vectors. So these viral vectors are modified, so they're stripped of their bad qualities, their virulence factors, but they still retain the ability to hold genetic material. So in this case, we fill the viral vector with our therapeutic gene sequence. This viral vector can then enter the stem cells of an affected individual, undergo DNA processing, and then enter this therapeutic DNA cassette can enter the nucleus and integrates into a patient's chromosomal DNA. And the two important things that result from this are expression of the therapeutic mRNA and ultimately protein, but also that this therapeutic DNA is now stably transmitted to all progeny cells. And so in theory, this should last for the lifetime of the individual. So even though there are now over 450 described causes of inborn errors of immunity by genetic disease, the current clinical trials of gene therapy for IEI sort of revolve around three categories. One is for SCID. There's 4 trials for SCID. There's been 4 products developed for SCID. So adenosine deaminase deficiency, X-linked SCID, Artemis SCID, and RAG1. There have been 2 for neutrophil defects, including leukocyte adhesion deficiency type 1 and X-linked chronic granulomatous disease. And then 2 for other combined immunodeficiency, and this includes Wiskott-Aldrich syndrome and IPEX, which stands for immune dysregulation, polyendocrinopathy, enteropathy, X-linked. In terms of the studies that are actively recruiting, this includes the 3 in SCID. I'll note that the RAG1 study is only recruiting in Europe. And then still for X-linked CGD, and then a T cell trial for IPEX, a T cell gene therapy trial for IPEX. The other diseases are trying to open up further studies or are in different phases of development, with all of these with the ultimate goal of trying to put it on the market and make it widely available to all patients. And so if I focus just a little bit on SCID, what gene therapy has allowed us is to treat patients when they look well. And so as you may know, newborn screening for SCID is now being done in all states in the US. And so what this means is that we pick up SCID cases very early, and we can treat them with definitive therapy, either through allogeneic bone marrow transplantation or gene therapy with autologous transplantation when they look like this. So you can tell these are 2 patients who we treated at UCLA with SCID. They look like normal, healthy babies. You cannot differentiate them from any other baby. Instead of treating babies when they look like this. This is actually another SCID baby that came to us from Lebanon. He was 6 months old at the time when he came, but he weighed only 50th percentile for a 1-month-old. So he was very emaciated. He was not healthy looking. You can tell he kind of has this dusky color. He was very unhealthy and stayed in our ICU for quite a while. Fortunately, he's doing now very well after having received gene therapy. But ideally, we would like to treat them sooner before they get to this stage. I would also like to point out that even though viral vector-mediated gene therapy has been life-saving for many individuals, there are still some pitfalls that we're trying to address. So the first thing is that the expression of the gene is not normally regulated. And the reason this happens is because the therapeutic gene cassette, when it gets integrated into a patient's chromosome by the viral vector, it's done so randomly. It goes into random places in the chromosome, and so it doesn't have the signals to tell it when to turn off and on. And so many times it may stay on, or maybe the expression is kind of weak, so it varies. And then the viral vector can integrate near a proto-oncogene and turn on its expression, leading to cancer. And what I mean by that is illustrated here is that there's a polyclonal transduction, meaning when we first treat the stem cells, many stem cells are treated, and the viral vector gets into many different places, but then there's ultimately a monoclonal expansion. So if you imagine that all these cells are your patient's bone marrow stem cells, and we treat them all with the viral vector, but it's not 100% efficient, so it won't get into all of them. Let's pretend it gets into these 3. And then it'll integrate in different places in the DNA for all 3 cells. So maybe one will have it, in this chromosome, one will have another chromosome, one might get 2 copies of the vector. And for simplicity's sake, we'll assume that the cells that don't get the vector don't engraft, or they don't survive, or they don't lead to production of immune cells. And so immune reconstitution is left to the cells that got the vector. However, if one of the cells receives a copy of the vector near a proto-oncogene, then that cell might have a a survival advantage or sort of a preference for higher rates of expansion. And so you get more of that particular clone, which isn't necessarily an issue until there's some sort of leukemic transformation in one of those clones. And that then that makes the replication of that cell go even faster and more awry, and that could lead to leukemia. And that is, in fact, what happened in many of the early clinical trials of gene therapy for inborn errors of immunity. So fortunately for ADA SCID, it only occurred in 1 in over 50 patients treated, but you can see that for Wiskott-Aldrich syndrome, chronic granulomatous disease, and X-linked SCID, that many of these patients developed leukemic events due to the gene therapy. Fortunately, we've been able to get around this and address this issue by transitioning from an earlier form of viral vector called a retroviral vector to more recent forms of viral vectors called lentiviral vectors. And lentiviral vectors tend not to integrate near active genes. And so that sort of decreases the chance of integrating near a gene that's actively transcribed and translated that might lead to leukemogenesis. However, these cases of leukemic events in these early clinical trials did demonstrate to us that maybe a more specific approach for gene therapy might be favorable. And so this has been enabled by what we call programmable nucleases. The most common are zinc finger nucleases, transcription activator like effector nucleases, and CRISPR-Cas9, which is really sort of exploded the field of gene editing. And so I'm going to show you an example of what we're doing with CRISPR-Cas9 and really focus mostly on CRISPR-Cas9, even though the other two technologies are still being used. And so in order to understand how these programmable nucleases work, we first need to understand what happens to our DNA when there's a break, because these programmable nucleases cause breaks in the DNA. So normally, if our cells are exposed to ionizing radiation or something that causes there to be a double-stranded break in the DNA, it can be repaired most easily through a pathway called non-homologous end joining. And I sort of consider this to be a Band-Aid where the ends of the broken DNA are sealed up and then quickly glued back together. And in that process, you're not going to fix anything there. So you're going to have lost or gained some little pieces of DNA. However, the benefit of this is it happens quickly and you prevent any further damage to the rest of your DNA. The other main pathway that can occur is called homologous recombination DNA repair. So this can occur if your cell where the DNA break occurred happens to be at a stage where it's getting ready to undergo mitosis, meaning it's getting ready to make a copy of its own cell. And when it's about to undergo mitosis, it has replicated all of its chromosomes in the form of a sister chromatid, because those will split off into the new cell. So when that sister chromatid is available and a DNA double-stranded break occurs, that double-stranded break will use the sister chromatid as a repair template. And it will look at the sister chromatid, match itself up to it, and actually fix the base pairs that are missing or that have been added. And so this is a base perfect repair mechanism. And this is, for the most part, what we try to harness for gene editing. So in the context of genome editing, this is what it would look like. So we would artificially create double-stranded DNA breaks where we want it to occur. So usually at the site of a mutation that causes a disease. We would do this using ZFNs, TALENs, or CRISPR-Cas9. So you can see that we created a break in the DNA. This DNA can be repaired either through non-homologous end joining, which could be helpful if you're trying to knock out a gene for some reason. However, if you provide the cell with a donor DNA, so what I'm showing here is the blue donor DNA is your therapeutic sequence. and then you flank it with what we call homology arms. Those purple homology arms mirror the area surrounding the cut site. So you're sort of tricking the cell into thinking, oh, that's my sister chromatid. And then the cell will then undergo homologous recombination using those purple flanking homology arms as their repair template. And then we sort of integrate the therapeutic donor DNA into the chromosome. And so gene editing is being explored for many IEI. I tried to list as many as I could here, but I know that there's more being done. But what I thought would be a nice illustration of how gene editing could be applied is our ongoing work for X-linked hyper-IgM or defects in CD40 ligand. So just in a very few brief slides, X-linked hyper-IgM syndrome is a genetic defect characterized by the inability to switch from production of antibodies of the IgM type to antibodies of IgG, A, or E. So in order for our B cells, which make antibody, to make class-switched antibodies, so this is IgG, A, or E, it needs to interact with a T cell through various co-receptors. But an absolute requirement is interaction between CD40, which is always expressed on B cells, and CD40 ligand, which is expressed on T cells only when the immune system is activated. So when you don't have proper expression of CD40 ligand, you have the X-linked form of the disease. And so patients present with a combined immunodeficiency manifesting as recurrent bacterial sinopulmonary infections, pneumonia. So 40% of patients present with pneumocystis jirovecii pneumonias, complicated gastrointestinal infections, including those with cryptosporidial species, autoimmunity, and cancers. We know from prior work that gene therapy for X-linked hyper-IgM requires site-specific integration. So these two very important studies that were done in the late 1990s and early 2000 were in mouse models of the CD40 ligand deficient disease. And so what both groups did is they used a viral vector to introduce a copy of the normal murine, so mouse CD40 ligand cDNA, into mouse stem cells. They put these stem cells back into the mice. The positive finding was that many of the mice reconstituted their immune system. So many of them were able to respond to immune challenges such as exposure to attenuated viruses or other immune stimulation. However, 2/3 of mice in that top study and 1/3 of mice in the bottom study developed abnormal lymphoproliferation. And the reason this occurred is because when you integrate the murine CD40 ligand using a viral vector, it's always on. But that's not what is supposed to happen normally. So typically in a healthy individual like you and I, when we're not sick, our CD40 ligand expression is probably 1 to 3%, probably less than 5%, quite low, because those T cells are not activated. We don't need to be actively pumping out new antibody. However, when we get sick or we get exposed to any pathogens, our CD40 ligand expression goes up very quickly, and that stimulates the B cells. And then even before our infection gets better, the CD40 ligand expression goes back down. And so you can see that you can sort of understand how CD40 ligand expression needs to be really tightly regulated. And so we can't just throw in the gene and always leave it on. So we really need a site-specific approach for this. And the way in which we would do this is to try to insert a normal copy of the CD40 ligand gene where it should go. Okay, so the way that we did this is we designed CRISPRs that bind to the early part of the CD40 ligand gene. This results in a double-stranded break that can be repaired through non-homologous end joining, which is not our goal. Or if you utilize the sequence surrounding the cut site as those homology arms that I talked about earlier, and you use those homology arms to flank a therapeutic human CD40 ligand gene cassette, you could allow homology-directed repair and integrate this therapeutic CD40 ligand gene under control of natural control elements. So it's in its natural location, and it now can receive signals as to when to turn on and off. And so we tested this in T cells from X-linked hyper-IgM patients. And so what I'm showing here in the series of flow plots is restoration of CD40 ligand expression. So you'll see on the left, when we stimulate T cells from a healthy individual, that there's a high upregulation of CD40 ligand expression. When we stimulate T cells from a patient, there is no CD40 ligand expression. And then when we stimulate T cells from a patient that have been gene modified and gene corrected, you can see that we restore a large percentage of that CD40 ligand expression. We also wanted to make sure that this gene that we introduced was behaving physiologically. And so the way that we did that is we looked at CD40 ligand expression at 1 week intervals, and we would stimulate them and let them come to rest. And so if the CD40 ligand gene was behaving the way it should be at rest most of the time, and then stimulate when we add certain reagents to stimulate it, and then come back down to rest when we take it away. And so I'll point your attention to the light purple dots, circles. Those are all the cells that are left to rest at 1 week intervals. So you can see there's low levels of CD40 ligand expression. And then the dark purple points that are up on top are all the same cells that are stimulated at 1 week intervals. And so we see this very nice pattern of resting, stimulation, resting, stimulation. And then finally, there's a clinical assay to look at the function of CD40 ligand. And the way that we do that is we expose it to a form of CD40. So that's CD40 ligand binds to CD40. And then we're able to pick up that signal using flow cytometry. And so on the left, in a healthy control, you can see that high pink peak on the right, where the activated T cells bind to CD40. And then you see a similarly high peak in T cells from patients that have been gene modified on the right. We also have been demonstrating the ability of CRISPR-Cas9 to allow gene editing in peripheral blood stem cells. So these are hematopoietic stem cells that we ultimately would like to transplant into an affected individual. Because we're very interested in the ability of these gene-modified stem cells to form T cells, since, as I shared in the beginning, that CD40 ligand is expressed on T cells, we utilized a system developed by one of our collaborators called the artificial thymic organoid. And what this does is you gene modify hematopoietic stem cells, and then you put them into this 3D culture along with a different cell type that gives that culture a little bit of structure. There's an air and liquid interface, and you kind of imitate the formation of a thymus. And what I'm showing in the series of flow plots in the left are sort of the development of mature T cells in the system. And on the right, in the bar graph, we see that we can still have good rates of gene integration in T cells that come out of the ATO. So the first bar is showing that our stem cells, when they're gene modified, have around 25% gene editing. And then when we put it into this ATO system and let them become T cells, that we have around 15% editing, which we would consider therapeutic. And so I'll finish this part just by showing a timeline of the gene editing work for X-linked hyper-IgM, because it really helps us understand why this work takes a long time. So you can see that we started this in 2012 when Talons had just come out, and then shortly thereafter CRISPR-Cas9 came out, and we started incorporating that into our work. There was a lot of funding that went into developing this. And in 2019, we had an interact meeting with the FDA to discuss our work, which led to further funding, and then our current translational work, which is taking all of our data and experiments that we've demonstrated how this is feasible, and scaling it up to patient relevant sizes and using higher quality material. So our hope is that we can have a follow-up pre-IND meeting with the FDA in the next year or so, followed by an IND meeting and an early phase clinical trial in the next 3 years. But I will say that this timeline is similar to what many timelines look like for development of gene therapies for inborn errors of immunity. And unfortunately, in our current state, we're only able to work on, every disease has to have its own timeline. It's hard to combine diseases. And so, I think recognizing which diseases can benefit from this and having as much research to go into it is very helpful. So in conclusion, the field of site-specific genome editing has shown tremendous progress in the last few years. This is ideal for modification of genes that are tightly regulated. The off-target effects and effects on HSC biology need to be fully explored before transition to the clinic. But gene editing has the potential to revolutionize modern medicine if used responsibly and ethically. So I wanted to thank you all for being here today. Thank my group, who does this research with me, as well as my mentor, Donald Cohn, and all the funding sources that have allowed this work to progress over the last decade. So thank you very much, and I'd be happy to take any questions.
All right. Thank you so much, Dr. Kuo, for that in-depth overview of gene therapy and genetic testing. We now have some time available for Q&A. Before we begin, please remember that everyone's treatment and condition is unique. The information presented during this session is not medical advice, nor is it intended to be a substitute for medical advice, diagnosis, or treatment. Always seek the advice of your physician or other qualified health provider with questions concerning a medical condition. All right, we've got some great questions so far. Our first question is, how do I find out if I am eligible for gene therapy?
Yeah, that's a great question. So I think one is, to ask your immunologist or your physician. Many times, because we all attend conferences and communicate about patients, we generally are quite aware of the clinical trials that are going on. But if you wanted to do a search on your own, a great way to do that is through clinicaltrials.gov. So you can type into the search term your disease and maybe the term gene editing or gene therapy, and all of the trials that are both active or not active or inactive will show up. And so you can sort of look through them, see who the principal investigators are, and you could always reach out to their coordinators or to the PI themselves.
Wonderful, thank you. All right, next question. I know you touched on this in your talk, but just for the audience to really bring the point home, can you explain the benefit of genetic testing in relation to gene therapy?
Yeah, so as I sort of alluded to, our ability to perform gene therapy or gene editing right now is to correct or add a single gene. And so one is we need to know what gene is causing an individual's disease. And so, that's where genetic testing becomes very critical. We have to validate that, that if we do gene therapy, that it would fix a person's disease. The last thing we want to do is do gene therapy and add a gene and it doesn't even do anything. The other thing is many times when patients are enrolled in our gene therapy trials, they undergo additional gene testing. And the reason for that is because we want to make sure that there really is only one gene that is responsible for their disease. There have been a few cases in which there was a mutation or a variant in a different gene that not necessarily worsened the immune deficiency, but that made gene therapy a little bit less effective. And so, we really want to know what we're treating, and we want to know that we're treating the right gene, but also that there aren't other confounding genes that may affect the therapy.
Thank you. I know in our last question, you touched on clinicaltrials.gov as being a great resource for where you can find out if you're eligible. So does that mean that clinical trials are currently the only way to seek out gene therapy at this point?
Yeah, so currently, yes, unfortunately. All of the gene therapy trials that I listed earlier in my talk, they're at different phases of development. Some of them are still actively in clinical trial. Some are trying to start a next phase of their clinical trial, and some of them are trying to develop it into a commercialized drug. And that's when it becomes widely available to anyone without being part of a clinical trial. So right now, there is no gene therapy product that is available commercially. Pre-pandemic, there was a gene therapy product available for ADA SCID, approved only in Europe, called Strimvelis. However, because of some commercialization issues and sort of switching back and forth between different companies, that isn't currently on the market, but is anticipated to be back on the market hopefully soon, but also only in Europe.
Thank you, Dr. Kuo. All right, next question. What specific genetic testing would you recommend for someone with primary immune deficiency and autoimmune disorders?
So there's many different ways of approaching it, and I think it can depend on where you're being seen and what resources are available at your institution. I can speak really only for where I work, and then for some of my colleagues. But a common thing that we do now a lot, really for our primary immune deficiency patients with inflammatory complications, is first looking at their genes using a gene panel that's enriched for inborn errors of immunity. I used inborn errors of immunity a lot because there was this change in our terminology. Instead of using primary immune deficiency, we now use IEI because we want to acknowledge the fact that immune deficiency doesn't just mean lower immunity. It could also mean aberrant or overly active immune systems. So we want to include that in our definition. And so there are now many companies that do gene panels, and some academic institutions do it as well. Gene panels, including over 400 genes, that will look at all of the known genes that cause inborn errors of immunity. That's oftentimes a really good place to start. And I think the results of that, in combination with your immunologic workup, can help your doctors decide whether or not you need further gene testing, whether that's a whole exome sequencing test where we now look at more than 400. We're now looking at all the genes in your body that are expressed, that we know that are expressed, or even further whole genome sequencing, which is now becoming more doable and a little easier to analyze with better software.
Thank you. All right, and our last question. So if I have PI or IEI, as we just talked about, should my children and family members get genetic testing?
That's a great question. So, I would say that genetics testing should start with you. If you have PI, you should get genetic, tested and see if we can identify something that explains why you're having, your symptoms or your findings. And if we do find something, then we can do a more targeted, panel on family members, especially if they're symptomatic. And I will also add that, many companies that do these gene panels, if they sometimes will offer free testing in family members if there's a gene identified that's known to be familial. So that could also be very helpful.
Wonderful. Well, our time together has come to an end. Thank you so very much, Dr. Kuo, for your time and for putting together this wonderful talk. It has been an honor to have you join us today.
Thank you so much for having me. Thank you.